Dear Jürgen, dear Charlotte, dear Jakob, dear members of the jury committee for the Karl Ritter von Frisch Award, dear colleagues, dear early career researchers, dear guests and friends, on-site and connected online. This award is dedicated to the life's work of Nobel Prize winner Professor Karl Ritter von Frisch, who understood the diversity of biological sciences as a unity. Karl Ritter von Frisch and Jürgen Heinze have a lot in common: a love of exploring nature, enthusiasm for social insects, and above all, a knack for detailed experimental research to gain insights into intriguing phenomena. Before I highlight the laureates' many accomplishments, I want to elaborate on this love of exploring the natural world, because I think that Jürgen Heinze's fascination for nature is the foundation of his successful research. Some little anecdotes might help to reflect his enthusiasm. During a big conference, I was told, on social insects, he was spotted outside of the conference venue with a pocket knife in his hand and a piece of wood from which he had just extracted a cardiocondylar ant colony. He was overjoyed. Or, to the surprise of other attendees, Jürgen Heinze would take out an aspirator, a pooter, during a pause at the conference and start collecting ants while other people chatted. He was not concerned with what the others might think, but was enthused about what he discovered. Jürgen Heinze is a careful observer of nature, a naturalist. Natural history is where the inquiry starts, with careful observation of individual organisms in their environment, and from there expanding into the various levels of organization, from the individual organism to species, to larger scale ecological interactions, to evolutionary patterns. Thus, a first observation can be the starting point for well thought out experiments with the goal of gaining a more comprehensive understanding of what is going on around us, leading us to general conclusions and fundamental principles. Many experimental biologists follow the reverse approach. A conceptual question leads to an experiment in which the concept is tested. Clearly, this approach requires a good match between the question and the chosen species. Whether the question is relevant to the system studied is again a matter of knowledge on the species biology. Experiments on social behavior, for example, need to consider natural group size, for example, and composition to allow meaningful conclusions. Jürgen Heinze follows both lines of inquiry in his research in a reciprocally illuminating way. He gets inspired by natural history observations, undertakes clever experiments, infers generalities from the results, and tests theoretical concepts on suitable study systems. Jürgen Heinze's main interests are ant societies. He excels in knowing the detailed natural history of the many ant species he studies. This knowledge allows him to ask incisive comparative questions about the organization and the evolution of ant societies. A large body of his research centers around conflict and conflict resolution and the evolution of alternative reproductive tactics in ant societies, where he has shed light on communication networks and into hitherto unknown tactics of queens and, very importantly, also on the kind of neglected males in the systems. He specifically focused on two ant taxa, Leptothorax and Cardiocondylar, with extremely diverse biology across species and intriguing polymorphisms in both sexes. Jürgen Heinze has explored many facets in these taxa, from colony composition, development and aging to aspects of sexual selection, local mate competition that strongly shape the group dynamics. And in a few minutes, we will get a glimpse into this fascinating world through his talk. Early on, Jürgen Heinze recognized the benefit from combining natural history with molecular methods. He established molecular and genomic skills in his lab, thereby tackling first population genetic and sociogenetic aspects of ant societies. More recently, he combines experimental manipulation with transcriptomics, for example, to determine the mechanisms that cause reproductive task differentiation in a clonal ant species. Likewise exciting is the phenomenon that highly reproductive queens live longer than workers, although theory posits that there should be a trade-off between investment in reproduction and longevity. Further, by investigating the role of recombination rate and transposable elements, his team is currently exploring the genetic mechanisms underlying the expansion success of an invasive ant species. All these aspects are an integrated part of a broader agenda, ultimately aimed at the holistic understanding of how ant colonies function, how they match their environments, and how they have evolved. As a group leader, first in Erlangen and since 2000 in Regensburg, he attracted many early career researchers interested in working on ants. But this was not a requirement. Assets to his group should complement his skills, not necessarily be his helpers at the ant research nest. He seemed to have recognized a naturalist's talent in people, and several highly recognized researchers have grown out of his lab. They are of high praise and describe him as a very supportive group leader, quiet, well-organized, and engaging. He reliably supports his students with valuable advice as to research questions, experimental setups, and with his enormous knowledge of the body of literature. He does all this without much ado. On top of it all, Jürgen Heinze has been very active in scientific societies and committees. Over many years, he was a member of the DFG Expert Commission, a member of the Senate of the DFG, and a member of the DFG Zoology Review Board. Since 2016, he has been a member of the expert panel for the Federal and State Excellence Strategy. In 2018, he became member of the Wissenschaftsrat, the National Science Council, whose members combine scientific excellence with science policy competence. His degree of service to science and society is outstanding. It is not surprising that already in 2004, he was appointed as a member of the National Academy of Sciences Leopoldina. Jürgen Heinze's service to the community also pertains to zoology as such. To explain this, I would like to come back to the motivation for introducing the Karl Ritter von Frisch Award in 1979. Back then, Martin Lindauer wrote to Karl von Frisch, "We are concerned that zoology is increasingly disintegrated into subdisciplines that are isolating themselves from each other and thus losing sight of the big picture. Through the foundation of this award, we would like to counteract such developments." Jürgen Heinze's engagement for a united zoology is evident. Together with Barbara König, he established the section group Evolutionary Biology within the German Zoological Society, which provided a platform on the national level for the growing interest in the field. This section has been very active ever since and is interconnected with other sections such as systematics, ecology, and behavior. His involvement with the International Society of Zoological Sciences is another example. He has served as a member of its executive board since 2008. The goal of the society is to unite scholars to promote zoology in an integrative manner and foster international collaboration. Integration of subdisciplines of zoology is also the aim of the journal Frontiers in Zoology, of which Jürgen Heinze has been co-editor from the very beginning of the journal in 2004. With this journal that is supported by the DZG, zoology has re-emerged as an integrative discipline encompassing diverse aspects of animal life, from genes to ecosystems. It is one of the most important outlets of zoological research, and Jürgen Heinze's constant engagement is part of its success. I hope that this condensed account of Jürgen Heinze's scientific achievements, his support of early career researchers, and his engagement in science panels made it clear that Jürgen Heinze is an outstandingly worthy laureate. Dear Jürgen, let me congratulate you with all my heart on behalf of the German Zoological Society on the Karl Ritter von Frisch Medal, which you so well deserve.
Die Deutsche Zoologische Gesellschaft verleiht im Jahre 2021 zum 20. Mal ihren Wissenschaftspreis, verbunden mit der Karl Ritter von Frisch-Medaille, an Professor Dr. Jürgen Heinze, den Preisträger in diesem Jahr 2021, für seine umfassenden wissenschaftlichen Arbeiten, die unser Verständnis des Verhaltens und der Evolution sozialer Insekten wesentlich erweitern, sowie in Anerkennung seines Einsatzes für die zoologische Forschung auf nationaler und internationaler Ebene. Jürgen Heinze erforscht Kooperation und Konflikt bei sozialen Insekten mit einem konzeptionell und methodisch vielfältigen Ansatz. Verhalten, funktionelle Morphologie, Kolonie, Entwicklung, Sozialstrukturen und Fortpflanzungstaktiken von Weibchen und Männchen werden in beeindruckender Weise verknüpft. Seine Forschung führt von der artspezifischen Biologie sozial lebender Insekten zum konzeptionellen Verständnis der Evolution und Diversität von Lebensgeschichtsstrategien. Der Wissenschaftspreis, wie in der Ankündigung bereits adressiert, wird gestiftet vom Inter-Research Wissenschaftsverlag Oldendorf-Luhe, gegründet von Dr. H.C. Otto Kinne, und dem Springer Verlag GmbH Berlin Heidelberg, Deutsche Zoologische Gesellschaft, der Präsident. Es ist mir eine große Ehre, Ihnen dieses Dokument und das damit verbundene Preisgeld und die Medaille indirekt zu überreichen. Und ich würde dafür Charlotte und die Laudatorin doch bitten, noch einmal hier hinter diese schwarze Linie zu treten, damit wir das auch auf Kamera bannen können. Herzlichen Glückwunsch, die Medaille. Dürfen Sie zeigen, das ist schicker.
Vielen Dank. Dear everybody, I would like to thank everyone who is involved with this procedure of awarding me this very prestigious prize. I'm very happy. I feel extremely honored, and I would like to thank everybody, the jury, the members of the DZG. I would like to thank Gabriele Uhl for this very flattering laudatio, and I would like, not least, thank all my collaborators who over the last decades have contributed to my research. Without them, without my mentors, without my students, without my colleagues, it would not have been possible to stand here and to give a presentation. Hopefully it works. Okay, so from my first slide, you can see that I have been interested in digging around in the dirt and looking for something interesting for a long time, for more than 60 decades. My focus of what is interesting has changed over these years. And I think the title of my talk is very nicely fit also to this environment, because it is about aging and reproduction in social insects. And the title of my talk is The Queens, the Workers, and the Grim Reaper. And I think this matches very well the Catholic Church. I tend to forget my very last slide, so I would like to start with my acknowledgements. This is a word cloud of all my co-authors over the last 20 years. And you can see a large number of very famous names, such as Bert Höldobler, Susanne Volzig, Sylvia Krämer, Judith Korb, and others. But in the context of my presentation today, the people in black are the most important. That's my former postdoc, Alexandra Schrempf. It is my present postdocs, Jan Oettler and Abel Bernardou. They have contributed to the research that I'm going to present to you. I should also say that I have not only chosen this topic because we are in a church, but also because this topic, aging in social insects, is also currently funded by research in a research unit by the German Research Foundation. Now, what am I going to tell you in the next 25 minutes or so? I will first tell you something about aging and senescence so that you know what you can expect in the future. I will then try to explain to you that social insects are ideal models to test evolutionary theories of aging. And I will then try to answer 3 different questions, which have already in part been addressed by Gabriele Uhl. Why do queens so long? How can they be both highly fecund and long-lived? And what controls different life expectancies of queens and workers? Now let's start with the first part. We all age and die. And the question is why? And if you have not experienced aging yourself so far, let me just explain to you what aging means in humans. According to Medawar, aging is a collection of changes that render human beings and also other organisms progressively more likely to die. This is very nicely illustrated in these 2 photos of the same lady, once with 20 years old and then 100 years later. And as you can see, and this is a citation from Obie de Grey, a famous biogerontologist, aging is a barbaric phenomenon that shouldn't be tolerated in a polite society. Now, aging in humans is associated with many changes in physiology, in anatomy. We have a gradual reduction in height, loss of muscle and bone mass, a gradual reduction in various body functions. There's farsightedness, and for 50% of us, there's also menopause associated with aging. In addition, there's an increased incidence of pathologies, such as cancer, heart disease, arthritis, etc. And this makes it more likely to die at higher ages. So aging essentially means that we have an increase, an age-related increase of the mortality rate. And after 30, for humans, the chance of dying doubles every 8 years. This is nicely illustrated by Gompert's mortality function. But it's not only humans that age. All organisms age, but they age at very different speeds. To give you examples from plants, this here, the upper slide shows you the oldest non-clonal organism. It's a great basin bristlecone pine, and it is assumed to be about 4,750 years old. On the other hand, we have plant species such as Arabidopsis thaliana, which complete their whole life cycle in 6 weeks. We have almost similar variation in animals. There's an Antarctic sponge that is known to live for more than 1,500 years. And there are other Methuselahs like the ocean quahog or goldfish or red sea urchins, etc., which also live for several hundred years. On the other hand, a gastrotrichon completes its whole life cycle in just 3 to 21 days. Now you might say, okay, a gastrotrichon is quite different from a bowhead whale, but we find similar or almost similar variation also among very closely related animals. For example, the yellow rockfish lives for nearly 100 years, whereas the sockeye salmon of similar weight and similar size completes its life in only 5 years. So all multicellular organisms age and die. The question is why? And why do organisms age so differently? Just a few days or more than 1,000 years? Now, this is not an easy question. And in fact, it is a central question in evolutionary biology. And there are more than 300 theories that explain why we age and why we age at different paces. Many of these theories are only of limited applicability or plausibility, so they match only for a particular species. But there are 2 sets that are more widely accepted and can be more generally be used. And these are first, the damage-based theories of aging, and second, the evolutionary theories of aging. Now, damage-based theories of aging, I think that's easy to explain. If you don't repair damage, sooner or later you will fall apart. And this damage may come from damage to the DNA, somatic mutations. It may come from oxidative stress, that the free radical theory, and it may be due to telomere attrition, that is, the caps at the ends of our chromosomes shorten during each replication cycle. Now this leads to damage, but there are powerful countermeasures, DNA repair mechanisms, there are antioxidants, and there's telomerase, which elongates the telomeres again. So all these damages can be repaired. And I think, therefore, that the damage-based theories of aging are not sufficient. And here come the evolutionary theories of aging, which say that aging and death are necessary byproducts of evolution. There's a mutation accumulation theory, the theory of antagonistic pleiotropy, and the disposable soma theory. Now, the mutation accumulation theory suggests that there is a selection shadow at high age. And this is because most organisms die from external causes at young ages. So they die from predation, from diseases, or from the lack of resources, and they don't reach high ages in nature. And this means that the strength of natural selection decreases with age, and there is no selection against late-acting negative mutations. And when you keep an animal in captivity and allow it to survive longer than it would in the nature, then you will see that somehow these negative mutations come into action. The animals get cancer, or they get arthritis, or whatever. Now, some of these genes may increase the fitness of young individuals, but have negative effects later in life. This is the idea of an antagonistic pleiotropy. So a gene has two sides, and I try to illustrate this with this picture puzzle, where you can see either a young lady looking backwards, or you can see an old lady looking downwards. So the chin of the young lady is the nose of the old lady. So essentially, this means that a gene can have positive and negative impact depending on age. Now, the third theory is the disposable soma theory, and it is based on the idea that all organisms have only a limited number of available resources. And they can take these resources either to put them into maintenance of their own body, in repairing body damage, or they can put these resources into reproduction. So a balance needs to be found between the costs of maintenance and the costs of reproduction. As you can see here, you can either have a lot of young, or you can invest a lot in your own body. So this leads to a trade-off between lifespan and reproductive success. And this trade-off has indeed been demonstrated in Xenarhabditis worms, in Drosophila, and even in humans. And to give you just another example from cockroaches, here also we find that individuals that produce a lot of young early in life do not live as long as individuals that do not produce a lot of young. So there is a negative association between reproduction on one side and lifespan on the other. Now, all these theories are tested in a number of model organisms, which are short-lived and not very social. So they are very different from us. So this is why I think that looking at social insects might help to better understand why aging occurs. So social insects are ideal model organisms for aging research. You all know social insects. So certainly you know that there are honeybees, there are wood ants, etc. And you also know that cooperation and division of labor in these insect societies has led to a new level of selection, which has been referred to as the superorganism. So insect societies compete as individuals with other organisms. And this cooperation and division of labor leads to an enormous ecological success of these social insects. You certainly know that 30% of the animal biomass in rainforests is made up just by termites and ants. And also in other ecosystems, terrestrial ecosystems at least, social insects are extremely important in pollination, in predation, in direct and indirect herbivory, etc. So there are so many reasons to study live social insects. Why should we study aging and death? Isn't it only frustrating? No, it's not, because we have two very interesting aspects here. First, queens, the female reproductives, and in termites also the male reproductives, the kings, may be extremely long-lived for an animal of such a tiny size. So ant queens and termite queens may live for 30 years and more. This has been nicely illustrated by Laurent Keller and Michel Genoux in a paper in 1997. So the hatch bar shows you the life expectancy of solitary insects, and the gray bars show you the life expectancy of the queens of eusocial insects. And you see that there is a very big difference. Keller and Genoux interpreted this as evidence that external mortality shapes longevities. And we will later see that this explanation, though it's looks very obvious, is not the right one. The second reason for why we should study aging and death in social insects is that there is enormous variation among very closely related individuals. For example, Lasius niger queens, they may live for many years, and workers, which have the same genome, they live only for a few months. And at the same time, the queens are highly fertile, whereas the workers are more or less sterile. So this turns the fundamental life history trade-off between longevity and fecundity upside down. So the disposable soma theory appears not to match here what we find in ants. You might say, so what? Queens and workers differ in morphology, in physiology, in ontogeny, behavior, nutritional status. Why shouldn't they also differ in longevity and fecundity? However, we find similar variation also in species where we do not have any morphological or ontogenetical differences between reproductives and non-reproductives. For example, in the ant genus Diacama, mated reproductives and unmated non-reproductives are identical in morphology and ontogeny, and nevertheless, they differ strikingly in longevity. And what's more, in the ant Platytheria punctata, unmated workers produce females from unfertilized eggs by Thelytokous parthenogenesis, and all individuals are clone mates, genetically completely identical. But if you randomly choose a worker and allow it to reproduce, this worker will outlive all its non-reproductive nestmates. So the reproductives are the black bars here, and the white bars are the lifespans of the non-reproductives. So taking this all together, this leads to the already mentioned three questions. Why do queens live so long? How can they be both highly fecund and long-lived? And what controls the different life expectancies of queens and workers? And to answer these questions, we used our pet ants, Cardiocondyla. Cardiocondyla is a genus of about 80 species, and the individuals are very tiny, 2 to 3 millimeters long. And also the colonies are very small, less than 100 workers on average. And this is an advantage for aging studies, the queens and the workers are relatively short-lived. They live for less than 2 years, and the colonies can easily be maintained in the lab. So we set up experimental single queen colonies of various species, in particular Cardiocondyla obscuria. We had queens coming from identical rearing conditions. They had a similar genetic background, and we measured queen lifespan, we measured total reproductive success, and we looked also at weekly egg laying rate. And what we found is that in these single queen colonies, queen fecundity and longevity appeared to be correlated. So highly fecund queens live longer than less fecund queens. And at the same time, they produce more male and female sexuals. Okay, now you might say, this is trivial, because if you have more time to lay eggs, if you live longer, then you can also lay more eggs. However, it's not really trivial, because we also found a positive correlation between lifespan and the maximal weekly egg laying rate. So indeed, it appears that the highly fecund queens, the fertile queens, live longer than the less fecund queens. In addition, we found that in Cardiocondyla obscuria and other species, egg laying rate tended to increase with lifespan, even when we held worker number constant. So apparently, in contrast to most other species of animals and plants, we do not find reproductive senescence here. So how does this match the original idea that external mortality plays an important role? The idea goes back to George C. Williams. He said that higher adult death rates select for earlier senescence and shorter length of life, and this means that extrinsic mortality causes senescence. However, Murad and others have pointed out recently that this is a wrong interpretation. In fact, external mortality can also select for short lifespan. What is more important, according to Hamilton, is the proportion of newborns that are produced by parents that survived to a particular age. And here we have a study by Luisa Jaimez-Nidio and Jan Oettler. They looked at the proportion of young queens that are produced by mother queens of particular or of different ages. What you can see in different colony sizes is that old queens produce the lion's share of female sexuals. Queens produce most sexual offspring late in life. Now, from this study, we can draw these curves here. The red curve shows the relative mortality, and the blue curve shows the relative fertility. And why did I show you this? Well, there's a very famous paper in Nature, Jones et al. 2013, and they investigate relative mortality and fertility in a number of different organisms, animals and plants. What you can see is that in cardiocondylar ants, and presumably also in many, many other ants, we have a very different pattern. In particular, very different from what we see in Cinorhabditis or in Drosophila. So it's obviously a very different interrelation between mortality and fertility. It essentially means that we do not have a selection shadow, but that in contrast, selection is strongest on individuals of old age. And this has been very nicely shown in a recent study by Mark Harrison and Jan Oettler and others. They showed that in cardiocondylar obscuria, purifying selection increases with age, in contrast to Drosophila. So we can probably answer the first question, why do queens live so long? Well, it's late life fitness, which is important, as shown by Luisa and by Jan. And it's not external mortality risks, in contrast to what Keller and Genoux have suggested. But how can this be? How can queens be both highly fecund and long-lived? Well, obviously there is no trade-off for individual queens concerning reproduction versus longevity. And this has been very nicely demonstrated by an experiment done by Alexandra Schrempf. She manipulated egg-laying rate in cardiocondylar queens by removing daily, over many, many days, all the eggs from their colonies. And in this way, she forced the queens to produce more eggs than they would do in a normal colony. You can see this. The egg removal leads to an egg-laying rate of about 2.5 per day, and the control colonies, they had an egg-laying rate of about 1.5 or even less. Now, you would expect from standard models of aging that the manipulated queens live not as long as the control queens. But this is not the case. So this manipulation does not shorten the lifespan of the queens. So we obviously do not have this very famous trade-off. So it appears that we do not have any individual cost of reproduction. There is no evidence for the disposable soma theory, at least in our ants. Now, the queens are not alone. There are workers surrounding them, workers take care of them, workers feed them, etc. So there might be a social facilitation. Is it that the workers bear the costs of reproduction by taking care of the queens? This is not the case, because we see the same phenomenon in isolated queens, which do not have any workers around them. So queens which were highly fecund in isolation lived longer than queens which did not lay many eggs. The explanation for this is just another question. It appears that there is a mechanistic link between fecundity and longevity. And here I have a question mark, but later in the end of my presentation, I'm going to remove this question mark. But this question leads us to the final question: what controls the life expectancy of queens and workers? Now, You've seen that lifespan differs very strongly between highly fecund and less fecund queens, etc. But lifespan appears to respond very flexibly also to other traits of the social environment. For example, queens randomly chosen to be the only queen in their colony outlived queens in multi-queen colonies. And this was regardless of queen-worker ratio and without any visible queen-queen antagonism. We also see that worker lifespan is depending on the environment. So when we subjected 12 weeks old workers to the situation of colony founding by adding them to a new queen, they survived as long as freshly eclosed workers, which did not get a new queen. So it appears that in these old workers, the internal aging clock was set to 0. And what we have also seen is that colony size leads to different life expectancies in workers. This has already been shown by Olaf Röpel a couple of years ago in honeybees, but we could show that also ants live longer in small colonies than in large conspecific colonies. What does this mean? It appears in very simple words that ants live as long as they are needed. And this reminds of a hypothesis that was invented by Skulachev many years ago. He suggested that there's phenoptosis, that is programmed individual death, which is analogous to programmed cell death, apoptosis. This is highly controversial, and I leave it to the theoreticians to find out whether this could really happen. Kin selection might facilitate this. But if Skulachev was right, might there be genomic pathways that control fecundity and lifespan in response to the environment? Well, in insects, we know that nutrition, fecundity, and longevity are proximately linked through juvenile hormone, vitellogenin, and the insulin IGF-1 signaling pathway and other genomic pathways. And if these pathways are upregulated in solitary insects, then we have a decreased somatic maintenance, that is shorter lifespan, but we have increased fecundity. Now, it has been shown in honeybees that here these pathways are connected in a different way. So if we have an upregulation of these pathways, it leads to both an increased somatic maintenance and an increased fecundity. We investigated whether we find something similar also in our cardiocondylar ants. We compared the transcriptomes of young queens and old queens, and we found that more than 800 genes were differently expressed. And luckily, there were quite a number of orthologs of Drosophila genes that were involved in metabolism and in reproduction, etc. Interestingly, the direction of the regulation with age was very different between cardiocondylar queens and Drosophila. So what you can see on the y-axis is the change in regulation with age in Drosophila, and on the x-axis, we have the change of regulation in cardiocondylar. And in the square on the upper right, we would have genes that are upregulated with age in both Drosophila and cardiocondylar. And on the lower left, we have genes that are downregulated with age in both species. But what you can see is that there are quite a number of genes that are upregulated with age in Drosophila, but downregulated in cardiocondylar and vice versa. If we look at GO terms, we see that, for example, genes associated with neurogenesis or anatomical structure homeostasis are identically upregulated with age in both taxa. But there are quite a number of GO terms where we have an opposite regulation. This is not only on the level of GO terms, but also if we look at individual genes, we see, for example, neurolazarillo here, that is upregulated in Drosophila and downregulated with age in cardiocondylar, etc. One gene that I find particularly intriguing here is the insulin receptor gene, which is upregulated with age in cardiocondylar, but it does not change with age in Drosophila. Now, why do I find this intriguing? Well, in the research unit 2281, which is led by Judith Korb, it has been shown that this insulin receptor and many other genes in these various pathways are important in quite a number of different social insects, be it termites, be it bees, be it ants. In particular, juvenile hormone and genes associated with production of juvenile hormone turned out to be extremely important here, as shown in a meta-analysis by Judith Kopp. When we look at Cardiocondylar obscurio again, we also see that genes that are associated with juvenile hormone biosynthesis are upregulated with age, and genes associated with juvenile hormone metabolism are downregulated with age. So this again points to an involvement of this TIJ life network, as Judith has referred to this whole picture, but it appears to be in a different way than proposed for honeybees, in particular because the insulin receptor gene appears to be upregulated. And with this, we come full circle to the first slide again, because this insulin receptor gene is referred to in Caenorhabditis as the Grim Reaper. So essentially, we came full circle to my first slide. Just to summarize, why do queens live so long? Well, I think it's due to late life fitness, not external risks. How can they be both highly fecund and long-lived? There's a mechanistic link between fecundity and longevity. And what controls the life expectancy of queens and workers? Well, we know that there is an involvement of nutrient sensing pathways, insulin receptors, and juvenile hormone. Social evolution, I hope I could convince you, affects the pattern of senescence and longevity. Fecundity and lifespan are positively associated in social insects, and there is no evidence for strong trade-offs. Lifespan appears to depend on the social environment, and this variation is presumably controlled via the IS pathway. Now, the story is not completely clear yet. The research unit is still working on it, but maybe someone else in a few years or few decades can tell you about what has come out of this research, which we recently started. I thank you for your attention.